Hey everyone, so today we're going to be talking about transcription, specifically in prokaryotes. So first off, I have a few slides of definitions, which I'm going to pause on, that way you can come back to, but most of these I'm going to be explaining later on in the PowerPoint. So first off, we have the steps of transcription. So basically we have initiation, where RNA polymerase locates and binds to the promoter region on the DNA strand, and then the double helix unwinds. Then we have elongation, which is where the RNA polymerase actually creates the RNA where nucleotides are added. And then termination, which is where RNA polymerase reaches a termination sequence on the DNA strand, usually a termination hairpin, and then falls off the DNA strand. And this is where transcription is finished. And so you can see over here, this is a double helix unwinding. This is kind of what it would look like before transcription starts. So here we have a DNA strand. We have two strands, which is the coding and the template strand. The template strand is the one that is actually being read by the RNA polymerase to create the RNA. And so as you can see here, there are a few different regions here. This little circle is the promoter region. So this is the region that the RNA polymerase locates and then binds to. And then the -10 and -35 regions, these are regions. The -10 is the ta-ta box is what it's called. And so these regions are basically where it binds to the RNA polymerase. That way it knows the RNA polymerase holoenzyme binds to. That way it knows where to start. And then it starts actually transcription at this +1 region. And that's after the sigma factor dissociates. That way the RNA polymerase can actually move along. And so as it's going from this +1 site all the way to the terminator site, it's reading the nucleotides and adding the corresponding nucleotides that it needs for the RNA. And then it reaches something called the terminator. It's a terminator sequence. And then a termination hairpin, which I'll show you in the next slide what it looks like here. And it's basically just where the strand doubles back on itself, and then this causes the RNA polymerase to dissociate, so it just falls right off here, and then that actually ends the strand, the RNA strand. That way it's the correct length. And so I have an example here, which is what would happen if the ta-ta box, that -10 region, was gone when we're trying to create a sugar metabolizing gene. And so basically what would happen is that -10 box is very important. So if it's gone, then the RNA polymerase would probably not even recognize this promoter region. And so this gene would never even be made. And since this is a sugar metabolizing gene, this could be important for energy production. So that could be very bad.